Today I'm going to be covering DNA sequence solving and a couple of the key equations that we have seen so far and the type of problems that we may see on the exam. So I wanted to start with writing a complement to a DNA sense strand from 5 to 3 given a 5 to 3 sense strand. So sequence Z, shown below, is the sense strand. It goes CC, ATG, TG, TA, CGC, TTAA, GT, GAA, 5' to 3'. And this is the sense strand that I created to be given. And then in order to write it into a sense strand, the complement from the DNA strand, you would start by writing it backwards, which is just the reverse of sequence Z. And then once you have it written backwards, you write the base pairs for it, which gives you the complement strand to sequence Z. And thus you have the complement to a DNA sense strand. You could also do this in the reverse order, but this is just the way I found most convenient. You can also, and then I also wanted to cover writing an mRNA sequence strand from a DNA strand. Sequence Z is the same strand shown here, and written mRNA is essentially DNA, but with U's instead of T's. So all you have to do for translating DNA into mRNA is change the T's for the U's, and you can see that below in the other red writing. Another thing that I thought would be helpful to cover is translating an mRNA sequence into peptide sequence. So we have the codon table on the right, and then we have sequence Y, which is the mRNA strand from earlier. So essentially what you do is you take the 3 letters and match them up, and then you would write it out. And there are 3 frames that you can do this from, starting at the first letter of the sequence. Is the first one, PCVRLSE, and then the second one would start at the second C, which gives HVYA stop. And the third one gives MCTLK stop, and that would be starting at the A. So essentially you just take 3 in a row, look at the one in the codon table, and then write it down, and that's it. And then finally, this is just another add-on. I wanted to show calculating an estimated melting temperature for a DNA strand. So your sequence Z from earlier, each A or T pair is equal to 2 degrees for the melting temperature. Then each C to G pair, C or G pair, is equal to 4 degrees. So in the sequence above, we have 12 A or T pairs and 9 C or G pairs. 12 times 2 is 24, 9 times 4 is 36, 36 plus 24 is 60. So the estimated melting temperature for this DNA strand, sequence C, would be 60 degrees Celsius. Hope this all helps.